Our next speaker, I'm happy to introduce Maria Orive from the University of Kansas. I thought I would get a big round of applause because we have a big Who else from Kansas? Joe
Kansas.
Armin's coming too. He'll be here.
Yeah. Awesome. You might be asking yourself, well, first of all, I really love that introduction that we got, and it really helped set up why we might be interested in the price equation and thinking about multi-level selection, which was something you mentioned several times in the intro. You might ask yourself, why is Maria Orive standing there instead of, oh, I don't know, Andy, or where's Steve Frank, or maybe 5 or 6 other people in the audience. You'd have to ask the organizers what they had envisioned. But I took the term tutorial very literally. So we are going to walk through this like a good old-fashioned tutorial. So let's start with George R. Price himself. So in the 70s, 2 very, very influential papers that laid out what later was known as, we all call the price equation. And I just wanted to, I tried to find a happy photo of Professor Price, Dr. Price. He had a difficult life and a short one. And so take a moment. Okay, so let's talk about the price equation. And for this next little bit, I'm really leaning on, there's this beautiful, really nice paper by Andy Gardner, right there. Came out in 2020. And I like the way that this is framed there. So let's talk about the price equation. At the very core, the idea is the mapping of something, a trait or a quantity, between 2 aggregates. And many, many times the 2 aggregates are parent and offspring. And that's the terminology I'll generally use. But of course, it doesn't have to be. And the price equation gets used to explain a mapping of a trait between 2 aggregates in economics, for cultural things, for lots of other things. But we're mostly interested in biology here. And so we're going to focus on that. And I'm going to use this framing here. So what I've drawn here, and for some reason, this looks just like what Manus had. It's a circle with other circles. We have lots of circles. I'm not much of an artist. I'm a really terrible figure maker. So this is as sophisticated as the figures will be in this entire talk. So in the parent aggregate, I have labeled each parent uniquely. There's 7 of them. And then there's some offspring. And there's some trait that's varying that I'm following. And the trait here is the shade of that circle. If you wanted to think of these little individuals as little haploid, some little haploid creature, and maybe we're following a particular allele. There's, I think, if I've done it right, 5 different shades up there, and maybe then there would be 5 different alleles, A1 through A35. Okay, and then they have offspring. Just looking at this image, if I've done a good job, which I don't know that I have, you can immediately see 2 things about the process of mapping from this one aggregate to the other. And that's, well, the first thing is that not all the parents are represented in the offspring, right? So individual 1 doesn't have any offspring. Individual 2 has 1. Individual 3 has 2. So that's another part of it, is not only are not all the parents represented, but they could be represented by different amounts. So that's one aspect of this mapping that's important. But there's a second aspect of this mapping that's important, and that's that my trait, which is the shade of these ovals, and they might be alleles or something else, sometimes it matches between parent and offspring. Sometimes the mapping is sort of faithful. If you look at individual 2, it's kind of the one that goes all the way over. The shade of the parent and the shade of the offspring is exactly the same. But if you look at individual 7, they have 2 offspring. One's the same shade as them and one's darker. So that's another aspect of this. So we have 2 things. Whether or not the parents, sort of their representation in the offspring, that can be different. And also, once a parent is represented in the offspring, is it a faithful representation? So we will talk about that a lot. And I have notes because I'm nervous in front of this august crowd. Okay, so let's start defining things. I'm a population geneticist by training, and so I can't help but want to have PIs and things like that. So you're just going to have to sort of embrace it. So for this particular version of deriving the price equation, PI is the frequency of parent I. And we can imagine that in this particular case, each parent shows up one time. And so all of them, it's like 1/7. It's very simple. And we also are going to track a trait. And the trait's the shade in this case from white to black, shade of gray. My color vision's terrible, so I hardly ever use color as well. So we're only going to use black and white and a little bit of red just for spice. But we can imagine again that trait is instead a genetic allele. Maybe it's allele A1 through A5, something like that. Okay, we're happy with that. We are going to track in this particular version against this. I'll derive this a few different ways. Here, PI prime is the frequency of the offspring of parent I. So just as an example, parent 3, realize that might be a little small. I don't know if it's big enough for all of you to see. There's parent 3. Parent 3 has 2 offspring, 2 out of 7. So the frequency of the offspring of parent I in the offspring generation, if you like, is 2/7. There's many ways to define fitnesses, and we will, I'm sure, talk about that a lot over the next few days. But one way to define the fitness of parent I is the relative growth of its lineage. So we can define it as PI prime over PI. In this particular case, for parent 3, they have 2/7 of the offspring. They originally were 1/7 of the parent, so their fitness, that's a sort of a fitness of 2. We could also, if we want, scale everything to relative fitness. This particular case, of course, the mean fitness, the sum of all these fitnesses, the ways we've defined them multiplied by their frequency of the parents, just sums to 1, so it doesn't matter. The 1 is just going to fall out. Okay, so we have that. And then but we're interested in this mapping, and a mapping is sort of how does something change from one thing to another, right? So what we're going to do is ask a question about how does the trait change, the shade in this case. We're going to let Zi prime be the trait value of the offspring of parent I. If parent I has multiple offspring, it's the average across the offspring of that parent. And that's whatever value the parent had, Zi, plus a change, delta Zi. And delta Zi could be the average change in the trait across all the parent offspring. All right, so now so here in our little toy model, individual parent 2 has a delta Z of 0 because their offspring is exactly the same. And some of the other ones, it's not 0. Okay, now equations, but all the equations are going to be like barely math. It's my math. It's barely math. Okay, so we've got what we really want to know is the mapping of this trait from these 2 aggregates, from parents to offspring. So what's the change in the mean value of the trait, the shade? Well, that's the mean value in the sort of the offspring versus the parents. So we can go ahead, I can't help but want to point here instead of there. We can just go ahead and use the definition of the mean and the expectation, the frequency, times those values. We can use a little bit of definition here. I want to move this a little so I can see this easier. Sorry, I'm moving my Zoom stuff to another corner. Doesn't really help. Okay. What we have here is, again, that definition of fitness that I was using. So I can rewrite that so that the frequency of the lineage or the offspring of parent I, PI prime, is PI times WI. Again, this is our change in Z, and we can replace PI prime with that. And we can replace the I prime here, and we have it this way. We could expand it out to this. And then it's pretty clear, just looking at these 3 terms, that we have 3 expectations. The expectation of fitness times the trait Z, the expectation of fitness times the change in the trait, and then the expectation of Z itself. We can then just do that mathy thing where we just say, well, we really want to multiply by something, but if it equals 1, we can do that. Remembering that the way we've done this, the expectation of the fitnesses is 1. So we'll just multiply this by 1 in that form. Rearrange. Oh no. Okay. I have lost the ability to, maybe I need to do something here. Well, that worked. Rearrange things a little bit. And then, of course, now you see that these 2 terms form the covariance, and this gives this expectation. And this is price equation derivation number 1. You know when someone numbers their derivations, that there's going to be multiple derivations. And you're like, oh, why is that happening? That's happening. Okay, so this is derivation number 1, where we have the change in this trait that we're mapping from parent to offspring, is made up of two terms, a covariance and this expectation. The covariance is sometimes called the selection term. So the selection term is the covariance of the trait with fitness, right? And that makes sense, right? If we imagine that the trait, the darkness of my oval, covariates with fitness, say positively. I give more offspring if I have a dark oval, then yeah, I'm going to increase the overall value of the shade. It's going to be darker in the next generation. And then we have this other term, the transmission term, which looks at how the offspring differ from their parents, that delta Z. And now it's also clear, I think, that we're talking about those two things I said at the very beginning we have to keep track of in the mapping. The first thing, some of the parents will have offspring, some of them won't have offspring, and some of them will have offspring, different numbers of offspring. If that variability in having offspring is correlated, is related to the trait, then that's this covariance. So this term is taking account of that part of it. Well, the second part I said was, well, sometimes the parents and the offspring don't share the same trait. Sometimes the mapping's not faithful. That second term keeps track of whether the mapping is faithful or not. So those are the two things that I talked about, and they're shown up in this version. Just to keep us happy, remember I was assuming W bar was 1. You don't have to do that. And this is a real common way of writing the price equation, where the fitnesses are relative fitnesses, W or W bar. Sometimes we'll do that as well, but that's fine. And now an aside, two asides. Aside number one of the simple cases that comes up a lot in evolutionary biology is we say, well, the transmission is faithful. So now I've redone that drawing a little bit, and you'll notice that the parents and the offspring always have the same shade. We might imagine that this is a genetic model with no mutation, something like that, or some other aspect of transmission that's completely faithful. That's true. The delta ZIs are 0. That term goes away. The second term, the transmission term, goes away, and you're just left with that selection term, that covariance. Of course, we can rewrite covariance as lots of ways. One of the many ways we can rewrite it as a product of the variance and the regression coefficient, which is what's happening here. And then this is a version of the price equation that evolutionary biologists use a lot when they're talking about fitness surfaces in the very general way. So what we have here is a relationship between some measure of fitness and some phenotype Z. And let's say that that's the phenotype Z is a linear, the fitness is a linear function of the phenotype. That regression coefficient is positive, beta is positive. That would mean that the change in the trait would be positive. And so we often talk about sort of like, what do we expect selection to do? Do we expect it to increase a trait or decrease a trait? If we have a very simple model, this gives us what happens in that case. And then the second aside, and I feel like I have to make apologies to Steve now. Where's Steve sitting? I'm like, where are you? You're hiding over there. Yeah. Anyway, if you really want a better derivation of this, you should just corner Steve at the coffee or something. But one of the many places that the price equation kind of comes up is when we are talking about one of the ways to think about Fisher's fundamental theorem of natural selection, which is sort of here at the bottom. So in quantitative genetics, which I think we'll hear various talks over the week that are in that framework, we often define a phenotypic trait Z as the sum of two quantities. And in one of those quantities, which is this G here, is the sum of the additive effects on that trait, which is often called the breeding value. And then everything else, all the non-additive effects having to do with dominance, having to do with pleiotropy, having to do with environment, and having to do with interactions that are not additive, are lumped into this other term E. And if you focus on the additive effects, you only focus on G. If the trait that you're following, what if the trait you're following is fitness? What if fitness is the trait that you care about? Well, then you can do the same thing. You can decompose fitness, again, in this G plus E framework. And in the case of what, well, I think in the case of what Fisher was talking about, he doesn't actually derive it in 19, there's no equation, but I'm trying to interpret Fisher via Steve. He was considering the case where G is held constant, so that transmission term, delta G, is 0. And then we can just focus on the covariance and just focus on the additive bit, because this is not going to covary the way we've set it up with G. And so the change in mean fitness from one generation to the next should be the variance in that additive effect, which is the additive genetic variance. And so that's Fisher's fundamental theorem. And the fundamental part of it is basically saying, well, if a lot of the assumptions that we've made are true, that variance term has to be 0 or positive. So selection by itself can only change mean fitness for a population in a positive direction or not change it at all. And so that's where we get this idea that selection moves population sort of up a slope of increasing fitness always. But that falls naturally out of the price equation. All right. Let's do PopGen E. Let's just reframe this a little bit. Now my same little drawing of circles inside of circles, that's all there's ever going to be. Now we're following alleles. And I'm just going to follow haploids. I don't have to follow haploids. I could have done this with diploids. It gets messy to draw, but it's actually not that complicated to do. But let's follow haploids. We're going to have 2 types, big A and little a. I said I was trained as a PopGen person, so that's the only letters I like to use. And we'll have some average fitness of allele, big A, W big A, average fitness of allele, little a. P is the frequency of big A in the parent population, so we could have a mean fitness here. That's just the scaled frequency. The fitness is the average fitness is scaled by their frequencies. And then we can say, well, if the frequency in the offspring population is P prime, we're going to let that frequency be the original frequency in the parents multiplied by the average fitness of allele A over the mean fitness. And then this time, what we're going to track is the frequency of the allele. So that's the trait. The trait now that we're following, that's the mapping is that the frequency of big A. And so we want P prime minus P. I'll write that out. Fitness is a little different now. WI is the fitness of individual I. PI is the frequency of allele A in individual I. If you're not used to thinking about PopGen things, you may think to yourself, you said these were haploids. What do you mean frequency of an allele in a haploid? It's just 0, 1, right? So either I'm a big A individual and I have frequency 1, or I'm a little a individual and I have frequency 0 if I'm following my big A's. We can define mean fitness, we can define the allele frequency, and we can write the equation for the change in allele frequency, like we did before. Now we just do some bookkeeping type of math. We have that top equation. If we multiply both sides by the mean fitness, W bar, we get it in a slightly nicer form. And then if we just write in the definitions of the population allele frequency, the average fitness of allele A, population allele frequency again, mean fitness, and then we can do a little canceling and get this form. And this is obviously an expectation minus the product of 2 expectations. It's the covariance of the individual fitness with the allele frequency in individuals, whether they're a big A or little a. And what happened to my transmission term? There's no transmission term. It's kind of obvious, right? I said that they're little a or big A, and the offspring are little a and big A. And what did I not do? I didn't do any mutation. If I was doing diploids, which I could do, I could also say I have no drive, no meiotic drive, and no segregation distortion, so that my offspring are faithfully representing the frequency of A in the parent. So if I'm a heterozygote, my offspring faithfully represent my frequency of 0.5. So that's the assumption that's underlying this derivation. Okay, finally, we're getting to where we need to go, which is multi-level selection, which is where Manus told us to go, and that's where we're going. What if I add a level of aggregation? So I had an aggregate, another aggregate, but what if I have another level? So now I have subpopulations of my little haploid individuals within some larger population. And maybe this is the parent. I haven't drawn the offspring, but they're going to map onto. But the other thing I could do that might be more interesting, but it's harder to draw, is if my individual level is something like alleles maybe, and then my next level is diploid individuals with 2 alleles each. And then my population is a group of individuals. We can do that as well. Moving up and down that scale that we were seeing in the introduction, we can do that with this. It does just define which levels we're in. But we'll just have this abstract one just to let us derive something really quickly. So we have subpopulations that we're indexing with J. In each of those, the number of alleles, or the number of individuals, it's the same since we're haploids, is NJ, total population size of N. We'll track the frequency of A in each subpopulation, the overall frequency as well, P, and then we can track the mean number of copies of big A and the mean in both a subpopulation J and in the whole population. Do the same thing that we did before. We really want the change in the frequency of big A from one generation to the next. So using some definitions, using these to let us track what's going on, we can write out an equation for the change in the frequency of big A overall for the whole population. I've rewritten it here. We'll do the same thing we did before of multiplying both sides by the mean fitness W bar. And we will also use the definition of W bar to let us write it like this. And that's not so beautiful. I can't do a lot with that. I've rewritten it here because I'm doing that other little mathy thing that mathy people always do all the time, which is to add and subtract the same term and make things the way you like it. So I'm adding and subtracting this term because I want, you know what I want covariances, right? I mean, it's obvious where I'm going. But I do that and then I rearrange things. So I regroup these four terms into these little groupings. They're there again. And once I've done that, I can pull out of these bracketed terms a pj minus p and a pj prime minus pj. And you can see then, it's pretty obvious, that this quantity in each subpopulation is, I'm looking at that and it says weighted. It should be weighed, I think is the correct grammatical, but it's weighed by its relative size. The relative size of each subpopulation is nj over n, right? So that's basically an expectation. So now we have these two expectations. And if we remember that the expected value of the allele frequencies across the subpopulations is just p also, this ends up being the covariance of, this is important, the fitness in subpopulation J with the frequency of allele A in subpopulation J times this expectation. This is the fitness of the subpopulation J. That's important to know. Okay, now we're almost there. I just rewrote it again, and I've circled two terms in red, and they obviously have the same shape, right? So what if I just apply Price's equation to the second circled term? I can do that, right? There's nothing stopping me. So I'll do that. I'll say, well, in the same way that this is this covariance plus this expectation, the term that's circled is this covariance and this expectation, where here now the covariance is between the individual level fitness and the individual level frequency of big A, the zeros and ones, right? And this is at the individual level, again, fitness, and this is the transmission at the individual level. All right, if I expand it out, then I get this thing. This is the multi-level selection version of the Price equation. More commonly, it's actually this one. And it's important to realize what we've done to do that. We've assumed that the delta PIJs are zero. That's that transmission terms are zero and transmission is faithful at the individual level. So individuals are passing on, in this case, an allele frequency to their offspring faithfully. Yeah, that's fine. It's nice. And that is the common way. I've rewritten it at the top. In the second line, I rewrote the covariances in that way that you see really often, where instead of writing it as a covariance, you write it as a variance times the regression coefficient, because from an evolutionary genetics kind of point of view, that's often the way we think about with that regression coefficient. Is it positive? Is it negative? What's the direction of selection? If you write it like that, you can say, well, the multi-level Price equation then is separating out, remember we're making that assumption that transmission is faithful at the individual level, is separating out the change in the allele frequency or the trait, whatever is happening, into two pieces. One that has to do with selection between groups. Here they were subpopulations, could be something else, but it depends on the variation of J between my subpopulations and the relationship between my group fitness and the frequency in that subpopulation. That's what's usually referred to as group selection. Then we've got another term, though, and that's selection within groups, within my subpopulations. That depends on the variation in allele frequency of the individuals in the subpopulation and the correlation, the relationship between individual fitness and whether or not they're carrying that A allele, big A allele, 0. And that's often termed the individual selection. So this multi-level selection version of the price equation, separating out group selection and individual selection. And that could be really useful when we're thinking about conflict in different levels. And those levels could be different. Here it was these little haploid creatures sitting in a subpopulation, but maybe it's diploids, it's alleles inside of diploids, or maybe it's other elements, other genetic elements inside of any organism. Or you could move up and say, actually, what we're worried about is different organisms, the coral, the bacteria, and the algae, and the holobiont, right? It could be a lot of different things. The mathy part is the same. So is this helpful? Can we use the price equation to think about conflict? So I'm going to do that thing that people always do. Well, sometimes, maybe yes, maybe no. And I'm going to briefly talk about like sort of two vignettes that you're going to hear about much more about later. In fact, you're going to hear all about Hamilton's rule. I'm just going to show you how you derive Hamilton's rule out of the price equation, which I think is a kind of an interesting thing to see that connection, and then talk about something else as well. Okay, so first, maybe Hamilton's rule. So Hamilton's rule, which we will hear about much better explained in the next tutorial, gives us a way of thinking about helping behavior. In one version, it lets us think about helping behavior. And we could just think of it as like a really simple case. Let's say I have, I'm a PopGen person, an allele that has you help or not help. You know, if you have it, maybe you have helping behavior. And if you receive help, there's a benefit. But if you give help, there's a cost. And Hamilton's rule in its simplest form just says, well, the benefit has to outweigh the cost, just like all other things, to make things reasonable. But it's also weighed a little bit by how closely related you are to the person that you're helping. And that's Hamilton's rule. Okay, we're going to use that kind of framing to think about the price equation and Hamilton's rule. Let's just make the simplest possible model of sort of how helping behavior affects fitness. So WI is the fitness of individual I. And we're going to say, oh, it's just this additive case where it depends on W0, which is baseline fitness. It's a constant. And then two terms that depend on whether or not you help. There's a benefit, B, that's plus. But whether or not you get B depends on the probability that you receive help, that individual I receives help. I'm going to call that YI. And then there's a cost, and whether you pay the cost is whether you give help. This is really like very simple. Okay, if I do that, and this is this is the price equation. I'm going to get these messed up. The price equation, assuming transmission is faithful. Let's assume no mutation, faithful transmission. It's a big assumption. We can go ahead and replace the individual fitness by what I've written out, expand out that covariance, pull out the constants B and C from the covariance. But then the other thing we'll remember is this baseline fitness is a constant. So that covariance is 0. So the only two covariances we need to worry about are the other two. And then we can ask the question, well, if this is the framing for this helper allele, when will it increase in frequency? When will delta P be greater than 0? And it's pretty straightforward that it's when this term is bigger than this term, so when this inequality satisfies, right? When the benefit times that ratio of covariances is greater than the cost. And that already looks kind of Hamilton rule E, if only that weird covariance thing would be R. And so let's do a couple more steps. Again, we're gonna do things really simple. What if we have really simple genetics? How can we look at this? YI, I said, was the probability that I receives help, and I'm going to say it's a really simple function of the frequency of the helper allele in others, right? Because I need to receive help from someone else, from individual J. So that's PJ is the frequency of the allele in individual J. It's a 0 or 1. They're going to help me or they're not going to help me based on that. And then it's just this little linear relationship. And then also the probability I give help depends on my frequency of the helper allele. So it's do I have a 0 or 1? And that's PI. And then some constant that I don't care about. You'll see why in a minute. So then those two covariance terms are the covariances of YI with PI and HI with PI. And then using those equations, I can show that it's just K times this covariance or K times that variance. Okay, so I'll put those into the inequality I just showed from the previous slide. And then I get this term. And then that's obviously a regression coefficient. And now I've got maybe a slightly simpler version of when my helper allele might increase in frequency based on the price equation. That kind of intuitively makes sense to you, right? We've got the benefit and the cost, but we also need to know, hey, does this other individual have the helper allele or not? Like, because am I helping them? It's important to know that. Beta gives the relationship between me having the helper allele and maybe Mass having the helper allele, in case we're going to help each other get T or something later. There's very little cost to helping T, so maybe that one's not a good example. Okay, that's great. That makes sense. The spread of the helping allele depends on how well the allele frequency in the individual I, the focal individual, predicts the allele frequency in the other individual. And this is the same sort of idea that beta gives us that relationship. We need some kind of positive assortment of the helper allele between the two individuals. If beta is positive, that term might be. If beta is positive, it might be greater than C. It doesn't have to be. It depends on what the two of them are. But at least it's not negative. That's fine. When do we get that? Generally, we get that positive assortment when we have common descent, identity by descent, common descent. We share alleles through kinship or inbreeding. It doesn't have to. We can, I think today, not today, but throughout the workshop, we can talk about other cases where that's not true. And we can certainly think about types of aggregates where that's not the case. But we can sometimes have beta exactly equal R. Some mathy bits, but not very interesting ones. The question is, does knowing PI tell me the value of PJ? And it depends on whether or not the two alleles, in this case, the two haploid alleles are identical by descent. If they are, it tells me exactly. If they're not, then it's a bit random. So if they are identical by descent, then R is the probability they're identical by descent. And then this gives me, and then it's the frequency in J if they are. And then 1 minus R is the probability they're not identical by descent. And then I need to know what the frequency of the helper allele is if they're not. This one's kind of easy. If they're not identical by descent, the probability that some random J individual has the allele I need them to have, it's just the frequency of that allele in the population. That's just P. What about if we are identical by descent? Well, I could say that it's just the frequency in individual I. We're identical by descent, so it should be the same. And so if that's true, I can do a little algebra and boom, there's R, and I've retrieved Hamilton's rule from the price equation. I did do something not completely okay. I made this assumption, this one, that the probability, if we're identical by descent, the frequency in me and the frequency in this other individual should just be the frequency in that other individual. But that's sort of assuming we're identical by descent, not that, we are descendants from a common ancestor if we're identical by descent. So we're talking about a frequency in some ancestor and a frequency now. If selection is acting, that frequency can be changing quite a bit. If it's strong selection, it's changing quite a bit. And so it's possible that it's not the frequency of the other individual. So what I kind of did was I assumed weak selection, so that that's not a big change since the time when we were identical by descent. I think I hadn't thought about this before, but I guess it's also important to how long ago were we identical by descent? The more recently we were identical by descent, the less I think this assumption is bad. But if we weren't identical by descent since a long time ago, it's possible the allele frequencies are changing. I have to think about that. That just came to me. But nevertheless, we retrieved Hamilton's rule out of the price equation, and that's a connection, one of the connections between the two of them. I think it can be helpful to think about that. But I said also sometimes things are not so great. And I think, Manus, you were already talking about this. I feel very strongly that you were talking about this in my heart. I have no proof of that. So I was reading this paper in a really nice special issue of Phil Trans B by Shelton and Michaud. And they were talking about homogeneous groups. So what if I have clusters of individuals, but the clusters are homogeneous? Maybe because they are all descendants of a single cell, and there's no mutation or something. I don't, these, you could think of like, if you had no mutation, this can be like little multicellular groups. It can be all kinds of things. So inside the cluster, there's no variation. And so I just noticed that I switched back to Z from P. It's okay, doesn't matter. It can be P or Z, frequency or trait. The variance of individuals in a group is 0. These guys are all the same. These guys are all the same. There's variation across groups, but not inside of groups. Well, if I use the multi-level price equation, that means that this thing here is obviously 0. So that wipes this out. And I said that was the selection within groups. That's individual level selection. So it's kind of saying individual level selection does not matter for this. Is that true? Does no selection within groups mean no individual selection? I don't know. What if being in a group affects my fitness? It's not because I don't have a different fitness from the rest of my group because of some trait I have, but the fact of being in a group is different than the fitness I would have outside of a group, even though it's completely hypothetical. So I have no idea what my slide does next. It does this. Okay. So Shelton and Michaud talk about using contextual analysis and defining something called counterfactual fitness. They use the term they use the symbol omega for it. It's the fitness the individual would have if it were outside of a group. It's counterfactual because the individual is not outside of a group. This is sort of a hypothetical fitness the individual would have if they were part of some homogeneous group, because again, they're the same as their friends, that did not have group structure. All right, so that's the difference in fitness that being in a group causes, rather than the difference in fitness that groups have because the groups have different fitnesses. So it's not this thing, the variance, it's not having to do with the variation in WJ. It has to do with a difference in fitness of individuals due to being in a group or not. And they did something, I think there's another paper as well, but this is where I saw it. They said, okay, let's contrast the case of homogeneous groups with kind of where we would be with just the price equation and multi-level selection, and where we would be if we take this framework of thinking about fitness as having this part that can be due to being in a group, even if the group is homogeneous. And so at the top of this is the price equation again. Again, assuming this version assumes that the individual transmissions are 0. It's slightly simpler, but that's fine. So it had these two pieces, group selection and individual selection. And I've got my homogeneous groups. So for this, individual selection is 0. Price equation says it's all group selection. In this framing that Shelton and Michaud laid out, we can say, well, okay, maybe, but this is another way to do it. And I'm not going to go through their derivation at all. But we can think about 3 kinds, possibly more, 3 kinds of selection. Within group individual selection. That depends on within group variation in the trait, which doesn't exist here. So that's 0 here. So in this case, also 0. But then when we think about sort of a global sense of being in groups, we can separate out what they called specifically group selection from what they called global individual selection. So they define global individual selection as the covariance between this counterfactual fitness and the frequency of the trait value in the group. So if the trait makes you more likely to be in the group or not, whatever, and then that is this counterfactual fitness. If there's no positive effect of being in a group above what the group fitness is, then that term is 0. And you'd still have this, right? Specifically, group selection looks at what they call delta WJ. It's a little different from what we did before. It's the difference between the fitness of the group and sort of counterfactual fitness of the group. If this is 0, then this is just WJ, and this is just the covariance of groups with the frequency of the trait, or the value of the trait, and then these two things would be the same. So if there's no counterfactual fitness, these two things are the same. This is something separate that they kind of pull out. And again, mathematically, I didn't go through any of the derivations, and this is not my model, and I'm not sure I don't understand it super well, but I think it's I feel that already we've talked a little bit about whether using multi-level selection tells us everything. And I feel like there are cases like homogeneous groups where multi-level selection might not tell us everything we want to know, because it sort of says, hey, there's no effect of individual fitness, and yet we can kind of imagine that there is an effect of being in groups that in itself favors the individual or doesn't favor the individual, but doesn't have anything to do with variation inside of groups. It's the presence of the group themselves, and that's outside of this price framework. Maybe? I don't know. You have an entire 4 days to think about that. And that's all I have. Other than to say, when I was preparing this, there's so many papers on the price equation, so many papers, and they're awesome. And here's a few that I spent quality time with. There's many others, but if you want to play with those, you can. I wanted to thank the organizers. Again, mystifying why you chose me, but okay, that's fine. And I'm really looking forward to the talks because I feel like we're going to have a lot of fun. So that's all I had. Thank you. And we have, I think, several minutes to discuss, 15 minutes, maybe 10 minutes. It's like a talk show.
Okay, thanks, Maria. Yeah, I know you said that this isn't your model, and so
Not my model.
you know, don't feel the need to defend it too heavily. But when you introduce this counterfactual fitness, I think the thing that worries me is that then this W bar on the left is not the same as the W bar at the top, right? Because you're not decomposing those covariances or the covariance from the expectation. You're adding in this counterfactual scenario and then trying to equate it to the scenario you had before.
So again, not my model.
No.
It's like not my circus, but these definitely are my clowns. I mean, these are my clowns, obviously, but I think that these two things and this are defined somewhat differently in this model. And so W bar, you're right, is, well, no, I think W bar could still be defined the same way. It's just how you carve up the relationship between variation in groups and within groups, between groups and within groups to W bar. So I have a feeling that W bar isn't the issue. It's making sure you've accounted for everything correctly in these bins is the issue. Because this is not the same as that also. I mean, they're both 0 in this case, but this thing is also not the same as this. So they're defined a bit differently because I believe they've, this is not the price equation. You have not broken things up into different covariances. And, I need to, so this paper is, they have another paper where they work through the model, and it's very specifically, but it's cited in that paper, so we can go find it later. They're really specifically looking at an example, I believe, of paramecia, maybe? It's single-celled organisms, and they're really, really looking at sort of what the fitness in the group versus other group. Because they're looking at, not surprisingly, because you see Michaud's name on there, they're looking at the evolution of individuality and multicellularity from single cells. So I think you're right that there's more to this that I didn't talk about. I don't think it's the mean fitness. I actually think it's this decomposition over here is more complicated than I wrote. I believe that to be true. Yes, Professor West.
That was an awesome talk, Maria. That was really brilliant. So I don't know if I'm going to phrase this very well, but is this decomposition trying to solve a problem that's not there? Because the within group selection is not individual selection. It's relative fitness within groups. So like, I'm thinking of the sex ratio literature that addressed this in the 80s, like Steve's papers and things. You can have a trait which increases the direct individual success of an individual, but it's mutually beneficial. So it can if it benefits others, it will be favored purely because of it gives an individual fitness benefit, but it reduces your within group selection. So is there sometimes a confusion that just arises from calling within group selection individual selection?
Well, that is why I tried to be a little careful to not do that, except here at the end, to call it selection within groups rather than individual selection, because those are two different things. Here I sort of said, okay, no, let's call it individual level selection. I think when I was reading this and I was reading some other things, this idea of the problem with homogeneous groups, and I think it's really a transitions and evolution question, is that we sort of imagine the transition to multicellularity involved maybe groups of non-multicellular, unicellular individuals that were probably genetically identical, maybe genetically identical or identical. And so those homogeneous groups, how do you form a homogeneous group if there's no benefit to being in a group apart from the difference in my group versus another group? I feel like World Cup could be a good analogy, but I can't do it on the fly. But how do you do that? And I think that's the question they're trying to solve with this. And so maybe it's not hypothetical, but it's really rooted in that idea of that step from going from unicellular to multicellular or other kinds of groupings, that you start with a group that's the same. So there's no benefit yet to being, in a group. You're all the same. So there's no individual level fitness, but what if there's an advantage of just being in the group? And I think that's where this is coming from. That's my reading. And there's people here who probably know the answer. Yes?
I can just shout.
I think you have to be recorded, though. I think you have to be on the mic.
So this paper is new to me, although that's embarrassing because I had a paper in that issue. But it strikes me it does solve a problem. It solves a really old one. It's Williams' herd of fleet deer problem.
Oh, and they talk about that in the paper.
Okay,
but I just don't understand it beautifully enough to use it as an analogy, and I can't draw deer.
And that's been something that's been troubling for group selection decompositions of the price equation for years. And it strikes me that this solves it.
So this is the idea of, let's imagine that you have groups that are sort of not haphazard, but kind of haphazard. Like we have groups of deer that are fast and groups of deer that are slower, because some of the deer in the group are fast and some are slower. You could say, well, the faster deer are going to survive because something's chasing them. But they're not a group in that like everyone shares a common trait. They happen to be fast. And so there's an issue having to do with, if the group is not a true group that kind of reproduces itself, so there's a fitness of the group, you can get the same problem. And that they did bring that up in this paper. I just didn't use that analogy because it's both a little more complicated in my brain, and again, I can't draw deer. So good point. Yes. Yeah, you're just gonna run around. I'm not gonna do that. Oh, and then you.
I'm also not familiar with this, but I didn't have a paper in this issue. And what I'm a little confused, I guess it kind of should emerge from this, is why they're homogeneous. Is it because there's no gene part here? It's just you have these phenotypically. And I'm wondering, because you could have groups where you have kind of feedback mechanisms. You see this possibly in something like dictyostelium, where you have cross talk between cells and they kind of co-regulate. And then that group might end up homogeneous. Another group's homogeneous, but the outcome, the sort of phenotype of the group, is some complex interaction between the members of the group. So I don't know where like the homogeneous, does it matter that it's homogeneous like at the phenotypic level, or all the individuals did the same thing, even if the group's homogeneous? That's not a homogeneous group.
I feel like the paper may address that, but I do not know.
Okay.
But I think that's a good question. Like, why are they homogeneous? Why are these groups homogeneous? I think in the idea, in what they were talking about a lot in the paper, which is this transition from unicellularity to multicellularity, they are probably homogeneous because they share common ancestry. And they happen to be, but I don't, that's a good question. In the sort of what I was thinking, it's posited that we have these homogeneous groups, and we have not posited how they came to be. And that's probably important. And you're right, because you could start off heterogeneous and you could make yourself homogeneous.
Yeah, it's sort of regulate yourself to kind of a shared.
I feel like that is not what this is addressing. And there was a question in the back row. Sorry.
So just go quickly again.
Okay.
Sorry. I think it applies to more than just homogeneous groups, though, because in every group,
Probably.
the individual's contribution to that group counts as part of the group fitness when it's really the individual's contribution.
I think you're probably right. But this specific example, I pulled it from homogeneous groups because I thought it was simple. It's not.
Thank you. And following up on the last points, I guess it's never been clear to me in kind of a group selection, especially like a social evolutionary context, what exactly is the mechanism that we're positing by which there is there's a fitness benefit from being in the group? I think in the context of social sciences like economics, we usually say something like divisions of labor, right? But this appears to be the opposite of like homogeneity. So as you were saying, we assume that groups start out kind of homogeneous, but if the boon is a kind of division of labor, then like there's no kind of fitness benefit.
Yeah, I think it's very, so you have to, in this very simple model, right? Everybody's homogeneous. So the benefit is being part of a group. It could be something as simple as having less or more, I'm bad at physics, surface area. I'm a mass like this versus I have less. So it could be things, but I'm in a group, and so not every part of my surface is exposed to outside because I'm part of this group that's mixing around. Sometimes I'm in the middle and sometimes I'm outside, something like that.
Yeah, I guess like the broader question is like, does this kind of framework have an answer for the question of like, how does being in a group kind of change your collective selective trajectories? Does it create more heterogeneity or more homogeneity? And do we have kind of an empirical or any reason to posit one or the other?
Neither Shelton or Mashoda are in here. So I'm going to say, yes, it does, because in their paper, they say it does.
Beautiful. Thank you.
So yes, it does. There. As long as if this counterfactual fitness is not zero. So if counterfactual fitness is zero, we're back to the land of price, and we don't need this. But as long as there's a difference in my fitness as an individual, that has nothing to do with my trait. It just has to do with whether I'm in the group or not in the group. And so the reason it took me a while to understand what they were saying, but we're not saying that you could be outside of the group. It's a completely hypothetical being outside of the group thing, which is weird, but interesting.
So just to clarify, this multi-level selection approach and proceeding all has transmission term negligible, right?
You don't have to. You don't have to. You can absolutely, I want to go back to here, where we first derived it.
Yeah.
You can absolutely be here. You're fine. You're fine to be here. We wrote it like this because this is really the way you see it all the time. And it lets you have this versus that.
Yeah. So
it's clean, right?
That's essentially my question, because to me, how my brain works, the really natural way to model something like meiotic drive would be to not treat it like this multi-level way, but to put that in the transmission term.
Yes.
But you clearly favor this approach.
I don't favor. I do not. I have no favor. I have no favoritism.
Oh, this is what I wanted to ask. Why do you present it like this?
This is what is in all the textbooks. Yeah. So this is the canonical. Is that the right word? I'm looking at Andy. Is it canonical? Would you say it's canonical?
I'll use that word.
Oh, Andy would not use canonical. It's the common. It's the popular. It's popular. Okay. Popular. Vulgar. Yeah, it could be the vulgate. As opposed to canonical.
It makes classical Mendelian genetics.
Matches classical Mendelian genetics. This is true because if the trait that you are following is alleles and allele frequency, just segregation and Mendelian inheritance gives you this exactly because we don't have mutation in it. Thank you. Thank you.
But I feel like it is in the transmission term. In the first line, you got the expectation term, and that within group selection part is equal to that. So,
so there is a transmission, but that's the group transmission.
But if your group is like individual,
you just have to define your group properly. At the bottom level, at whatever your bottom level is, you've assumed it's faithful. But if you're, you could easily have meiotic drive. So actually, the diploids I didn't use. Maybe I should have used them. So I've got diploids. Let's say the bottom term is the alleles. And then I've got diploids. And then I've got individuals in a group. Now, the bottom term is the sort of fate of the allele. So I can say, oh, there's no mutation. But the middle term is me as a diploid. I'm a diploid. And so meiotic drive can be right there. It can be that transmission, and it's the delta PJ in that case. Thank you. I got us out of that.
Thank you.